Good morning, everyone. Good afternoon, and good evening. My name is Shigato Das, Director of Partnering at BIO. Welcome to What's Hot and What's Not in Oncology Licensing, a Google Hangout hosted by BIO and ShareVault, moderated by Linda Pullan with Jeff Bachman, Vice President of Defined Health, Paul Junowitz, Vice President, Oncology Business Opportunities at Sanofi, and Farron Pratt, Vice President of Strategic Industry Ventures, MD Anderson. Don't forget to join us at BIO Investor Forum in San Francisco, October 7th and 8th. If you're a biotech investor or in-licenser looking for your next find or biotech seeking capital, BIO Investor Forum is the industry-leading venue to have private partnering meetings, see company presentations, and panels on cutting-edge topics, and witness fireside chats with leaders like Ken Drazen of J&J Innovation. Now, I would like to introduce to you Steve Joseph, my co-host from ShareVault. Steve, over to you.
Shigato, thank you for the introduction, and in addition, our thanks to BIO for co-sponsoring today's web panel with ShareVault and for organizing this event on Google Hangouts. I also would like to welcome and thank the attendees at the event today. Our panel moderator is Linda Pullan. Linda is a well-known biopharma business development consultant with extensive experience that ranges from Pfizer to AstraZeneca to Amgen and others. This event is actually the 6th in a series on licensing that Linda has done with ShareVault. So Linda, thank you for the partnership, for suggesting today's great topic, and for recruiting the other panelists that the audience is about to meet. So with no further ado, let's get started with Linda and the panel who she'll be introducing. Linda?
Thank you. Welcome, everyone. Delighted to have you joining us. We've got a great session lined up for today. I want to also thank our hosts, BIO and ShareVault. As Steve mentions, I've been working with ShareVault for some time and have had a great relationship, and I think they really do bring a wonderful e-data room to the industry. And BIO, of course, we all know, and really everyone attends BIO meetings. I think for their BIO Investor Forum, it's a great opportunity for small companies to meet with investors. Well, let's get started on our panel introductions. Shigato, would you put up the slides on the panel members?
Sure.
We were fortunate today to have 3 great panelists that I did indeed choose with great care for both their deep knowledge and for their candor and directness, and I think we're going to have a lot of fun. Jeff Bachman comes from Defined Health. Defined Health is a leading firm providing strategic insights on opportunity assessments. Wow, that's quite an effect. On growth strategies, and Jeff has an encyclopedic knowledge of drug development and drug discovery, so I'm sure he's going to have lots to say. Paul Junowitz of Sanofi is responsible for oncology licensing at Sanofi and will bring us the real-world perspective of both having to assess many, many hundreds of opportunities a year and having to work with his internal team to figure out the strategic fit and the path for development. And so his perspective is clearly very valuable to all those who would like to understand how their opportunity is looked at. Farron Pratt is our 3rd panelist and a great addition. Comes from MD Anderson. He also has small company BD background before MD Anderson, but I think MD Anderson offers clearly one of the leading trial centers and cancer treatment centers, and I hope that he'll bring both his well-known candor and his clinical perspective of what's really important to patients. So with no further ado, we'll jump in and I'll ask a bunch of questions. Everyone in the audience is invited to type in their questions by going over to the left part of Google Hangouts and selecting the Q&A thing and typing in the questions, and we'll try to get to them both as we go and at the end. I've made far too many questions for my panel because I have lots I'd like to ask them. So let's get going and let's have some fun. So, first topic is approaches. And one of the questions that I get asked and I sometimes wonder about is how you panelists think about supportive care, anti-metastatics, and prevention compared to treatment of the tumor itself. Jump in.
You didn't start at ground level, did you, Linda?
Of course not.
I'm happy to go ahead. I mean, one thing I will say, I'll take the first shot here, and I want to specifically pick up on the comment about prevention, because I think, that is it's almost a verboten topic within industry because of the preconceptions of the challenges, the logistics involved, the time period for trials, the costs, obviously the risks also for patients because of therapeutic window. And the profile has to be very strong. We all know the history and precedence of the limited use of tamoxifen and other terms that have formal secondary prevention labels. So, and yet at the same time, we're in a situation where many in the field, oncologists and people studying what has classically been called chemoprevention, feel that this is where we need to go and should go, whether with some of the mechanisms that we have or other mechanisms that are more specific to the dysplastic or pre-neoplastic condition. So there are certainly lots of challenges, but I think that's a huge unmet need area. It's just, it's not something we're going to get to in the near term. So in terms of biotechs that might be thinking about working in the space, if you rank order those three, it's going to be therapeutics at the top, at least from our experience as kind of intermediaries and outsiders, with supportive kind of way down below that, and then way down below that, the idea of prevention.
So why supportive way below therapeutics?
Well, I think it's a function of both perceptions and misperceptions, right? So we had, for a while, oncology was really defined by those supportive care products, right? I mean, it was the ESAs and the GCSFs that were significant contributors in the kind of the early days of oncology, and that's not that long ago, really, right? We're talking about the early 90s, really, when the modern age of kind of oncology market started, and then antiemetics, et cetera. And as those have started to morph, go generic in some cases, et cetera, and with the rise of therapeutics, the kind of overall sense of where the value is, I think, from many of our, at least large pharma partners, is, most of them are not really looking at supportive, or when they look at supportive, it's always like, well, and also take a look at supportive almost as an afterthought. Specialty pharma, smaller pharma, and obviously some biotechs that think they want to bring something forward on their own still do ask us to get involved in looking at supportive. But I'd be very curious to hear, in particular from Paul, kind of from a large pharma viewpoint, kind of what they're thinking and what they're seeing from out there in terms of external opportunities.
Paul.
Thank you, Jeff. And I apologize. My mic isn't the best. In terms of supportive care, there's still a huge unmet medical need. And we always start with medical need. The problem is the science behind some of the new support care is very weak. So it's very hard to decide or determine the productivity of these anti-support care therapies. Sorry about the mic.
Good point. Ferran, did you want to add to that?
Yeah, our perspective is that industry in general is not interested in it, and that unless institutions like MD Anderson or Memorial or government institutions step up to it, nobody else is going to do it. So that's good news and bad news. Bad news because industry, I agree with Jeff and Paul, are not interested. Good news, there are other people that, for those out there that are listening to this panel, that have solutions in that space. There is hope, because there are people that recognize the value and their interest might be much more aligned with what they are trying to do.
But it's tough for the little biotech in that case, yeah.
Yeah, and I want to pick up on something. I want to echo that what Paul said about the unmet need. I mean, I think that the unmet need is really quite amazing. And in fact, I think there was a period of time where, maybe not insiders, but maybe even some insiders felt that, in the age of the targeted, agents, that we were going to be bypassing or dispensing with many of the side effects and issues that required supportive care from the days of cytotoxic, cytoreductive chemotherapy, when in fact that's not true, right? One, we didn't dispense with chemo foundation as backbone in many cases, and two, many of these cytotoxic, cytostatic, or targeted agents came with their own and sometimes new types of side effects that do need to be addressed. So I do want to underscore what Paul said about the need, but the disconnect between the need, the available valid or semi-valid even opportunities, and the lack of interest in kind of major pharma, which in part is dictated, I think, by what's actually available or looks like it's going to be worthwhile and available.
Can we take one example? Radiation-induced mucositis, huge problem.
Yes.
Can be limiting to curative radiotherapy, but we don't understand the mechanism. So we need to do more with institutions like MD Anderson to develop better models and better understand.
Great point.
Regarding prevention, we need to change the algorithm. Industry won't do it on its own. So we need to partner with academic cancer institute and the federal government. In my view, the NCI has not stepped up to what it can do in the prevention area.
Yeah, I would underscore that. It's almost one of those old jokes when at our annual conference, Cancer Progress, a couple years ago, when we put together a panel, because it's been a pet interest of mine in what is often called chemo prevention. It's almost the kind of session that nobody came to, right? I mean, it was very hard to get any industry representation. There were certainly some key and major seminal academics who wanted to get up there, but there was a certain aura to them of having been beaten down by working so long in the space, having interesting ideas, whether those are novel targets that are readily patentable, whether those are available, drugs or vitamins or other things that may have some value, and time and again having the door shut in their faces by industry. So it was kind of a frustrating session, one that I had hoped would have better attendance, and hopefully that will change. But I think what Paul said is spot on, that it will take a strong consortium between industry with government, and I think with agency, FDA and others as well, to really make something like that happen, short of some well-to-do person endowing and making a concerted effort through some foundation to do so.
Well, I think it's a great topic, but I think we need to move on to the next one for speed. So let's talk about various theories of cancer and how you think about them. Cancer stem cells, do you believe in it? Is it important? Is it different? Autophagy, telomeres, cancer metabolism, that's a good one. It's had a huge amount of hype, and let's start with those for the moment. Small question.
So some of these are more phenotypic than genotypic, and those are hard to target. So our focus is really on mechanistic, genotypic, and less on phenotypic targets. So epigenetics, yes. Cancer metabolism, yes. Autophagy, no.
I would add from the Andy Anderson perspective on what we see that's exciting. Epigenetics, everybody knows that it's hot, and yes, there will be a lot of advances, but cancer metabolism is coming of age. We already see several molecules that are much more promising than the ones that we saw in the past, that were toxicity was the issue there. But I think that we are at the cusp of getting very good cancer metabolism agents in the clinic.
Yeah, and I think, I think we know there's been a lot of historic controversy about cancer stem cells, and stronger evidence has for the most part resided until more recently on the kind of the heme malignancy side, and even not necessarily definitive there. But I think data are coming out, certainly stronger data about leukemia initiating cells on the heme side, as well as certain select cancers, whether glioblastoma or others. I think it's now more about identifying targets, and there certainly are biotechs with targets that are claimed to be sometimes solely on stem cells, sometimes between stem cell and bulk tumor cells, and that's reflected in their development plans in terms of whether it's a combination of being used up front along with the bulking, or whether it's some secondary prevention or maintenance setting. So I think we will be moving into the stem cell era, and certainly when you talk with KOLs, it's in their short list of places along with epigenetic and cancer metabolism. You didn't mention, because you probably want to reserve it for a whole separate section, immuno-oncology, but I think there's a intersection between immuno-oncology and stem cells, and even metabolism, et cetera, and that is kind of the whole tumor microenvironment, which if you take as a rich source of targets and interests between EMT and aspects of angiogenesis, let's move into there. I think that is a big area where there are more targets being identified, and certainly more interest, more overt interest amongst pharma, and so I think that's one you want to be sure to add.
And yet, anti-angiogenesis is a pretty tough area to partner in these days, right?
Well, because everyone's when mostly those are just kind of the 7th and 17th, anti-VEGF twist on the usual, which you can't do anymore. There was the time, as we all know, when just a novel target or even a novel platform to hit the same target with a slight tweak was sufficiently attractive to do partnering. But we know we're in a world now where you have to have some suggestion of proof of relevance and clinically meaningful differentiation, and it can be incremental in some cases, because there are certainly some cancers where any improvement would be important, but simply having a distinct mechanism or a next in class that doesn't really provide any value is a challenging place to be, and unfortunately too many biotechs kind of have followed down that path. I think Paul wanted to jump in with something.
Now, the other point is establishing proof of concept in the clinic. And you want to do that with small numbers of patients, 200, 250. If you need 700 patients to demonstrate that, like an angiogenesis, it's going to be tough.
Yeah.
Yep.
So we have an audience question, which indeed is about immunotherapies, and that's such a hot area. It's such a big topic. As Jeff said, it could be a whole session in itself. But let's go ahead and jump into that deep dive pool and talk a little bit about vaccines, checkpoint inhibitors, recruitment of tumor-infiltrating cells. Go for it, folks. Give me a little bit of flavor.
That's an area that I'm witnessing the frenzy as we speak.
That's exactly the
oncologists, as it's well known, there are people with relatively short attention spans, and now immuno-oncology is just so predominant that they barely think about anything else. And that's probably not the right place to be, but it's the reality. The frenzy on CAR T cells is something that it doesn't happen that often. But you see an authentic mania that companies feel that they need to be in, even though it's relatively early in the grand scheme of things. And in the past, things like siRNA, everybody also had the feeling that they needed to be in, and all of a sudden, everybody was divesting. It's tough to tell what's going to happen on CAR T cells, but the frenzy is just so big that immune checkpoints are considered old news. You see clinicians are still in the immune checkpoint. They still get excited by nivolumab and other PD-1 agents. We'll have to see what are going to be the best combinations. The jury is out on that, and whether PD-1 is really going to be. Say again?
And how many drugs in space can it be?
Correct. So vaccines are also considered a little bit of old news already. So yeah, I see the CAR T-cell space and the non-immune checkpoints, immunomechanisms of action being what's hot right now.
Yeah, so let me just add to that. I mean, I think that, the world can be chunked into, the areas that fit squarely within biopharma's bailiwick, which is, the antibodies, which is, the interest in checkpoint inhibitors, novel checkpoints, or, the eighth anti-PD-1, or in co-stimulatory agonists, is driven in part not only because of some very strong and in some cases surprising data, like anti-PD-1 in lung, but also because these are antibodies, right? Fit squarely within kind of the classic blockbuster oncology of the past, 10 or 15 years of the Herceptins and Rituxans, which of themselves also do exploit, parts of the immune system for ADCC for their activity. So I think that's why there's so much interest in those antibody approaches, because it fits very readily into the mindset. On the other hand, I think that the adoptive cell therapies is a whole other story, and the question originally started asking about TILs, tumor infiltrating lymphocytes, which I remember working on a project, in my early days in consulting at Defined Health, where it just was not viable because you had to remove the whole organ and then extract them, and then the results at the end of the day were not very good. So
we don't need any introducing.
Okay, very good. This is jumping ahead, but there's a couple of different audience questions that really talk about, okay, some of these areas are challenging, like being in anti-angiogenesis or other areas, and they're asking, so how do you really judge one early opportunity compared to another opportunity, and how do you think about what evidence of differentiation is enough? That's a huge topic and a great topic. Paul, you're a great one to touch on that.
Well, the way I look at it, Coca-Cola and Pepsi both do very well. But nobody knows RC Cola except for New Yorkers. So I think when you get to the fifth, sixth anti-angiogenic, it's very tough. You're talking about, subgroups of subgroups. So it's best to spend resources in new areas.
Yeah, I mean, unless someone was able to demonstrate, some really striking efficacy in true, say, bevacizumab-resistant patient setting, or clear activity in a setting where somehow Avastin had not been able to demonstrate. But what the chances of that, for a biologic, I think are limited, and we have some examples there. And the MTKIs are a whole other story. I mean, there continued to be failures even when one felt there was sufficient validation with those, where they were hitting in part, VEGF, PDGF, a number of factors involved along that pathway, or they're going for the very well trodden and very crowded space of RCC, where it's probably the last place one needs another oral MTKI at this point. Not to say that is well addressed. And frankly, there's still huge unmet needs, but what it needs is novel other mechanisms.
Mm-hmm.
Okay. So let me touch on one more modality or approach, which I know there are a number of young companies who are working in.
We lost you there, Linda.
Perspectives?
You're going to have to repeat that, Linda. You kind of cut out there.
Okay, shall I get? I was saying that that cytotoxics are an area where young companies work, but there's not, and they're aiming for better or safer cytotoxics, but it's virtually impossible to partner. And I was wondering if you share that perspective, that in phase 2 evidence of differentiation, cytotoxics are just unpartnerable.
I have my thoughts, but I want to let Paul go first.
Unpartnerable? No, but difficult. I mean, what's happened is people have used ADCs as novel ways of delivering cytotoxics. So I think cytotoxics are still alive, but in the form of a...
Right, I was thinking of that, yeah.
Yeah, and I just add to that I think I think part of the reason why people, so one would like to think that some of these younger companies are going after a next generation cytotoxic or a reformulation or a novel target even that would fall still into a cytoreductive cytotoxic chemo, category. Because they would be going after those because they had clear evidence that there was going to be some efficacy in some important setting and really add value. My more skeptical, jaded view from seeing many a small company is that it's driven more about, oh, this is a de-risk strategy. Sometimes they have in their mind that this is because there's a proven path for paclitaxel or for camptothecin, et cetera, that doing some slight tweaks, they'll have less risk, be able to get to market faster, et cetera. And frankly, when you look at the success rate of reformulations, they're bad. I mean, they're certainly no better than targeted agents or historical rates, and frankly, in some cases, they may even be worse because you only really have Abraxane and to a lesser degree Doxil to point to as successes in the field.
I would say that much like what we discussed in the very beginning, MD Anderson feels that it needs to help companies that are trying to bring cytotoxics because they are so difficult to partner, and because if we don't help, nobody else will. Much the same thing that we were discussing in the beginning. We're excited about them in the sense that we know that targeted therapy just can only do that much. And maybe we're being naive, but we still have the hope then there will come a cytotoxic, not only reformulation. We are more excited about new agents that will make a real difference. We still have that hope. When I came to MD Anderson, I thought that oncologists would be kind of turned off by cytotoxics, but that's not the reality. They instinctively believe in them. Nuking the system, there is something that instinctively appeals to them, and they think that there should be better tools out there.
Yeah, and I think there are some interesting targets that are being pursued. People are looking at different, not standard microtubules, but other, supportive structures within the cell. We go back to the history. Where did people first look at? They looked at actin, and of course, then everyone died. The models died. So I think there are lots of interesting cytotoxic targets. But again, I go back to the point that I said that one has to really demonstrate that these are providing a true value in the face of either a generic paclitaxel or docetaxel or doxorubicin, et cetera. So they have to be addressing a clear unmet need relative to what is often a generic, in part, if not total, chemo backbone regimen. So.
At some point, there'll be too many genetic mutations for targeted therapies. Cytotoxics will be needed. We will need novel cytotoxics.
Yeah, I think that's a good point, and the hope has been expressed for a while, and some studies tried to do it, of combine two targeted, quote unquote, targeted agents in the hopes of obviating the chemo regimens that they were being added onto. It hasn't quite panned out yet in most cases, but as Paul said, the more the more armamentarium we have. I think also we have a problem here because we're talking a little bit apples and oranges, because these are somewhat artificial distinctions. What is a targeted agent? Well, I would say that paclitaxel is pretty targeted. There's a difference between what its biologic target is and its effects, and whether it's cytotoxic or cytostatic. So there are all sorts of nuances here that are actually important when Paul's evaluating in-licensing opportunity, when Farron's thinking about what might come out of a lab that might be of interest to spin out into a company, etc. I'm trying to look at these questions too, Linda. We have a guitar.
Yeah, I do see questions on nanoparticles, which is it's a variation of what we were just talking about. That stuff, that's, I think, inherently difficult. I think that people like Paul have seen way too many of them. I would think that it's difficult to get excited by it. I don't know, Paul, if you agree.
Totally agree. However, we need to think about nanotechnology in detection of cancer, especially detection of relapse. Engineers are pretty amazing. So if we could really determine when cancer is progressing much earlier than radiologically, that would be a good thing.
Or to extend that even further, if we can detect it earlier in many situations where we're still detecting 75% of the cases when they're stage 3 or 4. Again, that ties into the original question you asked about, prevention, Linda. So, someone's asking here about, to go back something I was talking about they asked about the TILs when Farron was talking about adoptive cell therapy and CARTs. Can I just go back and touch on that to finish off, or?
Yeah, Jeff, I think that's a good idea.
So I think what I wanted to say was that the antibodies are in the kind of the sweet spot. Adoptive cell therapy is clearly a very hot and attractive area, as Farron said, particularly with the CAR T cells. I think the issue is that right now real proof of principle is really in ALL with CD19. So there's a lot of excitement, really perhaps getting a little ahead of the game for broadening that into other diseases, not just other heme malignancies, but also solid tumors. Also, these first-generation approaches are all autologous, ex vivo manipulated, and technology is moving so quickly that the 3rd, 4th, 5th, 6th generations are going to come very quickly. That certainly was, I think, in part behind the Pfizer-Cellectis deal, where they, proposed the ability to make, to allogeneicize, essentially make off-the-shelf types of CAR T cells. This would. And then, so I think the challenge with TILs, inherently is the degree to which they remain, autologous. I know I've seen some very interesting data from Cassie and Yi and others, but I think, it will still face some of the challenges that any autologous cell therapy will face in terms of having a higher bar of efficacy. And then also, ultimately, these are going to be competing against non-cell-based approaches like TCR and other multivalent approaches that try to do in the body, again, using that sweet spot of the toolkit antibodies to do what the cell therapies are doing. Now, whether they'll be as good as, I think that all remains to be seen. But
thanks, Jeff. That was a great summary. Maybe, Jeff, if you could look at some of the other questions and pick from them if there's one that particularly interests you or might be relevant to the group. Maybe we could continue with that. I think Linda might be having some connections with her, some problems with her connection, rather.
There is one question that asks, any particular areas where preclinical models or novel mechanisms of actions are likely to receive a receptive audience? One thing that is very hot, and I'm still a little puzzled by it, is all the bispecifics. I've seen companies reject antibodies on the basis that bispecifics are here and they are going to be better. And I'm a little puzzled by that, because I think it was you, Jeff, that was talking in the beginning. You were talking in the beginning about antibodies is what pharmas know, and it's something that it's true and tested. They know how to handle them and how to commercialize them. And I would agree very much with that. So I would like to hear from you, Jeff, and from you, Paul. What do you think about bispecifics, and why do you think they are so hot?
Paul, let's go over to you. What do you think about that?
Well, I think, again, the theory is they can bring multiple mechanisms to killing tumor cells. In some cases, this seems to work well. Blinatumomab is an active drug. It's likely to get approved in ALL. On the other hand, Genentech invested a lot in Herceptin bispecific without much, value at this point. So I think it's about the target. We should probably start with hematology, where the models are simpler, rather than solid tumors to establish proof of concept.
Yeah.
Great. And over to you, Jack.
I mean, I just want to say, first of all, that I think what Paul said about the heme models is is very true, and where we see a lot of biotechs. In fact, we see a disproportionate amount of our work over the past few years in the heme space, disproportionate to the epidemiology of the diseases. But on the other hand, they are very big market areas now. Compared to up there with solid tumors, even though they have much smaller demographics because of repeat dosing and multiple lines of therapy, etc. I don't want to go back to Farron's comment about the bispecific and what Paul said about it is about the target. I think that's very true. I think the bispecifics that people are more interested right now, and for which the data are stronger, are those that are really kind of combining, that are sitting within this immuno-oncology world, right? They're targeting a CD3 or something else, and some known vetted target, whether it's tumor-specific or tumor-associated. I think where it gets much more complicated, as Paul alluded to, is when you're trying to target two other categories like VEGF and EGF, right? I mean, a lot of experiments done independently in mice would have said, oh, that's a great combination. When the experiments were done separately with those programs, those didn't work. And so far, I haven't seen any strong data with a bispecific that puts those into a single molecule either, because I think there are challenges about, and we don't have the technology necessarily to do all the experiments to determine the right way to dose and time and sequence those relative to each other, because it may not be that you want to give them at exactly the same time or in the same molecule. May not be that easy.
That's a great comment. I'm sorry I dropped off for a little while. I lost my connection.
Welcome back.
Thank you. Let's talk generally about how one thinks about the ability to bring the story together and get your product assessed. How you think about the kind of data you need to see to be able to assess something.
So, again, I start with medical need. If medical need is minimum or unclear, we pass. After that, it's about target validation. Then the data have to speak for themselves. Lastly, we worry about commercial consideration.
Okay. So what's enough target validation?
Jeff?
All right. Well, I can speak for what we tell clients, which is just a surrogate of what I'm sure you're looking for, Paul. But one thing we say is that in our model systems for the even for the very youngest of biotechs, we and because sometimes they do have a plan of trying to do a deal very early on, but regardless, what we try and emphasize is you've got to have multiple models. Right? You've got to have models where you're adding on to standard of care as well as with an active control. You've got to study ideally in refractory populations, whether that's disease resistant to standard of care therapies, etc. And you've got to try to get away from just using the easy, very sensitive, models and cell lines that work with everything, right? Choose the harder ones, choose the primary, do the explants, etc., and so that the bar is much higher, and in fact that you don't get satisfied just seeing a slight diminishment in the growth curve, that you actually see significant flattening, if not regression. That's what we recommend.
Yeah, I was about to say exactly. I'm glad that you just said it, because the first thing that would come to mind is regression. The flattening of the growth curve, and probably you agree, Jeff, I don't think it cuts it anymore. I think that people want to see regression. Otherwise, I think it's difficult. It's going to be a question that's always lingering in the farmer's mind.
Regression is ultimate. Seeing no growth in a very large tumor is okay, but swelling in newly established tumors doesn't cut it. We also like to see a lot of DDX and target validate on human samples. Establish cell line or establish rodent models is insufficient.
Yeah, I want to emphasize something that Paul said too about we all know and seen those experiments time and again where it's introducing product X at the time of initiating tumor growth. So again, the idea of being able to regress established tumors, whether those are very large or otherwise, but at least showing significant effects on a very large and if not the regression in those that are less well established, as Farron and I had noted. So
those are great things, and I think there is an expectation these days that small companies will guide the potential partner to the best possible use. And that's often very challenging, right? But that is part of that expectation. Is that true?
It's true for us.
Yeah.
We try to give a lot of free advice. In the end, it's about the molecule. Everyone has good ideas. We just offer suggestions. But the real, intellectual property is in the molecule itself.
So we're running out of time, and we have many more questions than we can answer. Let's talk a little bit about clinical endpoints. And given the hotness of immunotherapies, response rates and tumor shrinkage are not their most important thing, and yet we still run most clinical trials going through a tumor shrinkage and then going to overall survival. Are we really changing the way we're thinking about clinical endpoints? And that brings in biomarkers, which is another huge topic that we could explore. Do you expect preclinical opportunities to have a biomarker when they're ready for partnering?
Yes.
I like that answer. Yes.
If you're really, focusing on a targeted therapy, you need a companion diagnostic or biomarker. If not, we're driving blind.
Right.
In terms of, clinical endpoints, seeing response is a true measure of anti-tumor activity. A prolonged, stable disease is okay, but tumor dependent.
I was just all I was gonna add to that was that I think that it's partially a function of the effect size. I mean, I think everyone wants to have a biomarker. There are some targets and mechanisms that are not so readily that's not so readily doable, at least not in a single individual biomarker. We all know the struggles that companies have had just trying to find some way to segment patients who are going to respond to anti-angiogenics. So it's kind of the ideal hope for situation, and at the very least, we tell our clients to kind of collect that information and that do those correlative and translational studies, so that even if you're somehow believing that you want to have a broad application of this, that may be inherent to the mechanism, that you still collect the information to allow you to see or to identify biomarkers if you haven't already pre-identified one. And it's also a function of the effect size, right? I mean, so many of these things are discovered kind of retrospectively when you don't hit the primary endpoint and you suddenly, identify, it's actually hitting ALK, right? That came after the fact because it was originally developed as an ALK-MET inhibitor, the crizotinib. So I think there's this interrelationship between, the mechanism, the ability to find that biomarker, the actual efficacy or effect size that you see when you use that. But I think the main point that Paul made is that, most pharma, if not all pharma, is setting that bar, where it's pretty much a given that you have to have some type of work or some demonstration that you're at least trying to identify that biomarker.
You asked Linda about immunotherapy and biomarkers for prediction of response. I don't see any of them in the near future. I don't foresee that a biomarker strategy to shorten clinical trials in terms of predicting who's going to respond, who's going to not, or to see early on who has responded, who hasn't, instead of tumor shrinkage, which is not the right one for immunotherapy anyway. I just don't see it. I think that for immunotherapy, it's going to be a long slog of looking at conventional measures of survival.
Yeah, I think the problem there, I mean, I hope that's not true, but I think for the moment you're very right, Farron. But I think the challenge is, and this will, aside from just the desire, the intellectual desire to try and identify those, or whether those are gene signatures or otherwise, I think there will be a very practical need, because we all know that these immuno-oncology agents are going to benefit from combination use. And if we're talking about a situation where just using an anti-PD-1 alone or an IPI alone is only having an effect in 20 or 40% of patients, depending on the situation, you can't pre-identify who those are, and then you're adding on a second very expensive agent, and you're increasing those that are responding, but you're not increasing it to 100%, so you still have a little bit of a crapshoot as to who's actually going to respond, and those responsive may go for several years. Being able to determine, who would be responsive and to really kind of determining the value there, I think is going to be increasingly important. It's a topic that will be front and center in our keynote at the Cancer Progress meeting next year in March. It's something that was a panel that head of our company, Ed Saltzman, chaired just this last week at Rodman Renshaw. And I think it's going to come up time and again with these very expensive agents that we know are going to be best used in combination. So if you can get a biomarker in there, that is really going to help everyone.
Let's talk a little bit about pricing and reimbursement. Certainly in hepatitis C, Gilead's had a tremendous amount of controversy for their pricing. In oncology, the Merck PD-1 has just priced and has scared a lot of payers. And yet at the same time, little companies look at those big dollars and say, hey, if they can charge that, I can too, can't I? And so how do you think about what the market will bear for a new treatment, and how a small company should think about price and reimbursement?
It's a key topic. If you don't get good pricing and reimbursement, your sales will be flat. So basically, one has to do some scenario based upon price and reimbursement, and then have a royalty or milestone based upon what you obtain. So if you really get good pricing and reimbursement, you try to obtain, and both parties should benefit. But you have to do this through creative business terms, deals.
Good point.
The news from the trenches here at MD Anderson are not encouraging. The noise about the price of drugs, fairly or unfairly, is very strong. It's an issue that it's top of mind for many for a lot of our clinicians. Some of them that actually have been quite vocal about it, even in the popular press. So it's an issue. It's not going to go away. It's going to become more and more a challenge in our view.
Yeah, and I would just echo that. And I think, you know, when we're doing assessments for biotechs where they're, you know, these are when the agents are at least in pre-POC, where we're doing very extensive forecasting and we're also doing market access and pricing work. You know, that type of work is not something that, you know, even 3-4 years ago we were doing consistently, right? We would do the modeling, but not necessarily market access. Now, we're almost always doing and building in that market access component, because it's not, although right now we're still seeing people slapping on, you know, a surcharge on top of the previous price of the drug that was in, but that's not going to be able to continue, particularly as they get combined. So again, this issue of demonstrating that value for the clinicians and for the payers is going to be increasingly important. And you know, whether that is aided and abetted by, you know, stronger data that is enabled by biomarkers, but certainly, you know, there are model systems outside the U.S. that, you know, while we may not go quite that far so quickly, you know, we are going to be in a different world 5 years from now. So we don't just look at benchmarking and saying, well, you know, we're going to add 10 or 20% to whatever, you know, an anti-PD-1 or something else was, because we just don't think that's going to be sustainable.
That's a great point. Well, we're down to the wire. What have I missed that you'd like to touch on? We have lots of potential questions. I think there's so many things we could cover, but have you each got one point you'd like to make as a closing?
I'd like to first thank everyone. My microphone has been a problem, but thank you. Cancer remains a major unmet medical need. We need to bring new and effective therapies for our patients. We need to work together on the business aspects and look for win-wins.
My last point would be something that we haven't touched, and it's about the role of diagnostics in oncology. Post-Prometheus and post-Myriad, the Supreme Court decisions in the United States, it's become exceedingly difficult to develop diagnostics in oncology. It's become almost an impossible task, and that's a real shame. At the same time, every threat is an opportunity. And the fact that this is an area now that is pretty much deserted might present opportunities for those that are brave enough and have the guts of bringing valuable diagnostics to the oncology field.
That's a great point. Jeff?
I guess my closing point will really be more that, you know, as Paul said, there's a lot of unmet need, there's a lot of hope, there's a lot of interesting novel mechanisms out there, and we've talked about some of the ways that companies have to make sure that they make their programs and those mechanisms shine in the best way they can, so that they increase the chances of having a deal. And the good thing is that for oncology, the trend has been to do earlier and earlier deals. There'll always be the big late-stage deals, but the trend has been much more for an upward increase in discovery and preclinical deals than in any other area. And those sometimes are still based on, you know, very hyped mechanisms, but by and large, you know, those are going to be, you know, where there is sufficient data and sufficient robustness of data and diversity of data to really support those programs. So to the degree to which these young biotechs can kind of spend the money and take the time to do it right, it's going to pay off in the long run.
That's a great comment, and I'll sort of use my closing remark to one thank BIO and ShareVault, and 2, thank my terrific panelists. But I'll pick up on what Jeff said, and I would encourage everyone to tap the potential great feedback you get from folks like Jeff and Paul and Farhan, and really,
you, Linda.
Thank you. I hope so too. And really think hard about how you design your program and communicate your messages in order to do those big, hot licensing deals. Sagato?
Well, thank you, Linda. I thought that was really great, and I would like to thank our audience for joining. This has been What's Hot and What's Not in Oncology Licensing, and we've been your host, Bio, along with our co-host, ShareVault. And we hope you enjoyed this. If you want to ask any more questions, you can use the Q&A feature, and what we will do is we will take the questions and coordinate with Linda to get selected questions answered. And I would like to thank Linda Pullan for being our moderator, Jeff Bachman, who was one of our esteemed panelists, Vice President of Defined Health, Paul Junovitz, Vice President at Sanofi, Head of Oncology Business Opportunities in the Strategic Portfolio and Management Group of the Oncology Division, Farhan Pratt, Vice President of Strategic Industry Ventures at MD Anderson, and our co-host, ShareVault. Thank you all for joining, and keep in mind this will be available for replay at YouTube, so please tune in to watch it again, and tune in to see an extensive discussion of this on Twitter.
Excellent.
Thank you, everyone.
Thank you,
Thank you
everybody.
so much.
Thank you, everyone.